Oliver. Thanks a lot. And I'm really happy. It's not lip service. I'm really happy to be here. I think this is just the crowd I would like to talk to, and this is to discuss with you guys. Because I came here with a wild hypothesis in my title, and with everybody I'm talking here, I have the feeling of, hey, they're sharing this hypothesis. So it might not be all wrong, so what I'm thinking, but we will find out. And I'm really looking forward to the discussion. So I'm going to take you back 500 million years to the Cambrian explosion. In those years, the average oxygen level in the ocean rose by 8%, and suddenly animal has exploded in terms of variations and numbers. And all life forms that still exist today came into existence in these years, in a slice of a couple of million years. Two great evolutionary biologists had studied the Cambrian explosion, and they came, in one aspect, to very different conclusions. Stephen Jay Gould, whom possibly many of you might know, said the following: if we were to rewind the tape of life and the same conditions were encountered again, evolution could take a different course. The human mind would probably never arise again, even if life's tape could be replayed 1,000 times. So what Stephen is saying here is that it's a completely random procedure. So the probability that things will move in the very same way is practically nil. It's completely random. Simon Conway-Morris comes to a completely different conclusion. He says, convergence hints at the existence of a deeper structure to biology. In this sense, the Darwinian mechanisms are a search engine to discover particular solutions, including intelligence. The evolutionary routes are many, but the destinations are limited. So what he says is there are boundary conditions in biology, some aspects that limit the variations. And they funnel the course of evolution in certain paths so that things can repeat itself. And our job is, if Simon is correct, to identify these boundary conditions. So what are they? So my question is, could cognition and its neural substrate have deeper structures, in sensu Simon Conway-Morris, such that they converge between species during evolution? So if he's right, we might see these repetitions all over again. And then we can start asking, what could be the boundary conditions? Are they on the behavioral level, and I take cognition as sort of behavior, or at the neural level, or the combination of both of them? And that's something I truly would love to discuss with you. For this, I take you back 324 million years. So in those years, there was a little animal living, West Lothiana lysée, you see it on the left. It's one of the candidates for the last common ancestor of today's mammals that you see there in the top, and today's birds that you see in the bottom. So we do not know if indeed this little lizard-like animal was the last common ancestor, but there was a small group of animals that possibly were the last common ancestor. To give you a hint what 324 million years imply and mean, I have to remind you of the following. About at 370 million years ago, the first fish crawled on Earth. So this is very close to that point in the evolution of vertebrate life on Earth. So it's a very, very deep bifurcation of the evolution of vertebrates. So when we compare humans and birds, we have a very deep comparison of two parallel groups of animals that lived and evolved over very long periods of time, parallel to each other. Ludwig Pedinger is one of my greatest heroes. He's a comparative neuroanatomist. He was the towering figure in his time, and he was exactly asking these kinds of questions. He wanted to reconstruct the evolution of the vertebrate brain, and by this, he was sure he could reconstruct the evolution of vertebrate cognition. In the years where this picture was about taken, he was pondering about a problem that was the deepest problem of his career, and that was the comparison of the mammalian and the avian brains. Let me introduce you to the problem Ludwig Pedinger had. His problem was the following. Mammals, he knew, had very large relative brains, so relative to their body size. Their brains were very large, and obviously also in those years, it was very much known about the mammalian brain. You see here in blue what we call the pallium. It's mostly cortex. I will call it isocortex here because it's more or less the same kind of cortex throughout the whole cortical tissue, and a little bit of hippocampus and amygdala. So the pallium was very large. Under the pallium was a subpallium, mostly what we call basal ganglia, and these are habit-based processes, motor reactions that are overlearned and automatically produced. At least in small birds, the brain was also relatively very large, about the same relative size as in mammals, at least in small birds. But their brain looked completely different. And for 10 years, he was trying to figure out what's going on in their brain with anatomical means, until he came to the conclusion that practically nearly everything of their forebrain was pallium, and nothing of that pallium was cortex, but it was all amygdala and hippocampus, and the rest was subpallium. So the conclusion at the cognitive level was these are robots. So the term robot was not invented then, but these are automatized systems that have highly automatized behavior without any cognitive ability. And there's a very moving passage in his paper from 1900 in which he said, now looking back, I cannot understand why he needed 10 bitter years to understand what is open to the first sight through the microscope, in that birds have no isocortex, but enlarged subpallium. I am one of those who demonstrated that Ludwig Eding was wrong. His whole theory was wrong. And I feel a bit bitter about that because I admire this guy. But what in 100 years people will think about Oliver and me, we don't know. But we do what we can do in our lifetimes under the conditions of knowledge that we have. So the verdict was very clear. No cortex, and that's it. So why is the cortex so important? It's very simple to show this. You take a rhesus monkey cortex, and this is an old depiction from Ludwig Edinger's times. And what you have in the cortex are two streams that are orthogonally organized. One is the vertical organization. These are the fibers that come from the thalamus. And just to give you an example, if it's coming from the auditory thalamus, this arrow would be, let's say, 300 hertz, 301, 302, 303, 304, and so on. An extremely precise projection onto the cortex. And along that red arrow, along the lamina of the cortex, there are specialized neurons that code for any aspect of the loudness of the sound, of changes in the sound, etc., etc. Orthogonal to that are the associative fibers. They connect everything with everything. Imagine a machinery that has an incredibly precise projection of everything that goes on outside of your body and from within your body. And it's able to associate with learning-related synapses everything you ever encountered with every other thing you ever encountered. The perfect machinery for cognition. And what a miserable brain is this. It has no cortical tissue, no lamination, no orthogonal orientation of canonical circuitries, and it has a second problem, and that is it's small. I told you that bird brains are relatively large. That's true, but it's only true for small birds. The larger the birds get, the less their brain grows proportional to their body size. So there are two problems. I'm dwelling on the second now in a minute. No cortex means no organization in the sort of cortex of cognitive properties, and small size. Let's talk about small size now. Let's take a chicken and a human brain, compare their size. It's a bit unfair, 2.7 against 1.3 kilogram. Human is a big animal, chicken is a small animal. Let's take a bird that is the largest bird that exists today, which is the ostrich. Male ostriches are a bit taller and way more than an average human. Okay, that's not that convincing in terms of brain size. Let's go for the moa, which went extinct about 9,000 years ago in New Zealand, a very large bird. Okay, Let's go for Dromornis, the largest bird that ever existed. It's 123 grams. So what you clearly see here is birds can grow to humongous sizes, but their brains don't grow proportionally. If you look at a very large mammal, for example, the orca, it's 9.3 kilogram of brain. So the conclusion is very clear. Even in large bodies, bird brains stay small. Mammalian brains can grow very large. The conclusion is equally clear. They have no neocortex, and they have a small brain. So that's a very dim prospect for avian cognition. And indeed, for a century, everybody was absolutely clear that birds are just colorful, tiny, cute little robotic animals. And when I started my PhD, I already had this hunch of there's something wrong with it, because that's not what I see. That's not what the most specialized literature tells, that nobody reads outside of the avian realm. And then later on it exploded. We now know that corvids, for example, have analogical reasoning, episodic memory, mirror self-recognition, tool use, planning, conceptual thinking together with parrots at the level of great apes, at the level of chimpanzees. But the bitter side of that is they just have 10 grams of brain. If you compare an animal without a cortex and 10 grams of brain, and it achieves the same cognitive properties as a chimpanzee with 400 grams of brain, you suddenly realize there's something wrong with us. So why do we need such a big brain to churn out the identical cognitive properties? And I will show you now one example of my own lab. Always when you see a person here with a name, then it's from my lab. When you don't see it, then it's from colleagues in other labs. So for example, mirror self-recognition, I will show you Gertie. Gertie was a magpie, he was just in mold, therefore he looks a bit unhappy, but he was very happy, I can show you. And he has a sticker here, a paper sticker, as in this picture. Birds have laterally placed eyes, so there is a mirror here. He doesn't look that way in the mirror, but that way in the mirror because of the lateral eye. He looks into the mirror, sees the sticker, then tries with all means to remove the sticker. It's then slided back sideways, and then finally he gets rid of it and is happy. So let's look at Gertie now. Life can go on. So the control group was not magpies without a sticker, but magpies with a black sticker. So the tactile aspect was identical, and they simply didn't care about the sticker then. So it was really visually triggered. So now our friends and colleagues in primatology were not that happy with these kinds of results. Because imagine a facility with chimpanzees or orangutans costs close to $1 million a year. It's a very, very big investment. But you can always argue that this is the only way you can study the evolution of complex cognition. And now there are all these people coming with animals just cost $20, and they do the same. That's a competition that is difficult. So they argued, not possibly because of competition, but because they deeply believed it, that we tested the animals always in that part of cognition in which they excelled. Well, primates have this breadth of cognition. So I sat down together with my friend Thomas Bugnia, and what we did was we singled out 8 areas of cognition that were so much widespread across the literature as possible: self-recognition, reasoning, object permanence, metacognition, theory of mind, mental time travel, and one reviewer forced us to also include vocalization learning, which we did, and practical. So the procedure was very simple. In each of these areas, we searched for the complete literature that was conducted with corvids and or parrots and monkeys and or great apes. And then we took experiments that are virtually identical. They cannot be identical. They're very similar as we could get it, and looked for the results, and looked whether there was a systematic effect in this literature between birds on one side and primates on the other side. I show you this for corvids, parrots, monkeys, and great apes for the exchange task. This is one of the tasks that we singled out because there are a lot of things we know. This could be the exchange task for me. And very unfortunately, I like sweets. So you could give me a sweet on a plate, and I would have to stare at it without being allowed to eat it. And the question is, how long can I stand that? Because I know if I stand it very long, it will be replaced with a baklava, which I would certainly prefer. How long can these animals do that? Obviously not with sweets and baklava, but because things that were previously then tested with differences in their affective properties. Ravens do it 6 minutes, great apes, gray parrots 15. Okay, that's not very convincing. And that's 4 minutes for chimpanzees. Here is a clear plus for the birds. We also find in some tasks things that are more pro-primates, but we could not identify a single property that showed a systematic difference between primates on the one side and corvids and parrots on the other. After this, 2 further reviews were published, one with 31 different cognitive properties tested in the same way. There was simply no systematic difference. So we can be pretty sure in saying, obviously there is no IQ test for animals, for non-human animals, but we did whatever we can to convincingly say we have no systematic difference in cognitive properties, but we have a major difference in brain size and brain structure. How do we explain this? But it's getting worse. You think it's worse, it's getting worse. It's not only the peak performances of the animals, but it's also the way they think is virtually identical. So many years ago, we had magpies that we were allowed to catch as nestlings from the nest, and they were then part of our family. We had to feed them every 20 minutes for 24 hours over several weeks. Please try to do that with your personnel without informing the unions of the Personalrat of your university. It's a heavy duty. But then in the end, you're rewarded with animals that really take you as their parents and as their comrades. So what we tested was object permanence. If you do not know what object permanence is, this is a mouse, you see it, you don't see it anymore, but you all know that object permanence, that the mouse still exists, then congratulations, you have reached level 4. I'm sure you all have reached also level 6. The introduction of this was happened by the famous developmental psychologist Jean Piaget, and he tested it and developed it with his own daughters. So what he came up with is a description how step by step his children moved up in the understanding of what we call object permanence by different kinds of tests. And it describes with incredible detail that when they, for example, are at level 4, what kind of mistakes they make when he gives them level 5 tests. How they reach level 5 then at one day suddenly, and then they fail at level 6 tests and what kind of mistakes they make, etc. And interesting, for example, if you take that and hide it behind you, and they've reached level 4, they look behind you. But by a trick, if the mouse had disappeared, they look behind you, and there is no mouse. They walk away. In the moment they reach level 5, they say, Daddy, what do you do there? Okay. You moved on to the kitchen, isn't it? I guess it is in the kitchen. So what do they do when they reach level 4 or level 5, before and after? So we took this toolbox in an identical way and adapted to magpies. Not only that our magpies also reached level 6 as humans, Not only that they reach level 6 earlier than we humans do, but they made the same mistakes. They took the same steps. Everything was identical. In the end, we said, if Jean Piaget would not have taken his daughters like magpies, he would have written the same book. So you have 10 grams of brain, you have no cortex, and not only the peak performances are identical, but the algorithmic processes and the development of these algorithmic processes are identical. But it's getting even worse. So I took out one more paper, and here we, with our colleagues in New Zealand, we taught our pigeons British orthography. Okay, so it's an inflated word. They learned properly written 4-letter English words to discriminate them from improperly, as wrongly written English words. So, for example, the word done. If you just swap, for example, in the middle, the position of the O and the N, then it's a non-word. So they had to discriminate words from non-words. Obviously, they got it on monitors, let's say done on the one side and a non-word on the other side, and they had to decide which one to pack. In the beginning, they had no clue. They just were random. But then miraculously, they get better and better and better and better. And then at one point, we put away all words. We had several thousand that we ever had used, and came up with a complete new set of words and non-words that the animals had never seen. And spontaneously, they reached performances, let's say, between 70 and 85%. I would say they might have passed second grade primary school, not with perfect grades, but they would have passed it. Okay, I'm not commenting on this one. So it's sort of a school, it's a circus trick to show that they can. It's become science when you discover the mechanisms. So by analyzing the data in great detail, we came to the conclusion that what they used were two strategies. They looked for the average bigram frequency. So the bigram DO has in British language a frequency of 0.032, ON has 0.007, and NE has 0.026. The average bigram frequency is 0.022, which is a high value. So when the combination of these symbols looked associated with past rewards, they packed on it. When it's very strange looking, they went for a non-word. So Levenstein distance, you possibly know much better than I do, is a different version of something remotely similar. So they use these two strategies. That's exactly what we do when we glance through a paper and then say, oh, there's a typo here. So it's just a frequency-based, very simple-minded rule that the animals used, and that's exactly what we do. So again, they use strategies that are at least similar to what we do. So we are faced with two major problems here. And I tell you, I'm not going to solve them. I make them worse and give a few hints of what the solutions might be. Birds learn, remember, forget, err, conclude just like mammals. So we have two problems. Some avian species, corvids and parrots, reach the same cognitive abilities, but with non-cortical brains of 3 to 25 gram. Chimps having 400 gram. So how is that possible? Second, they show the same functional properties, the algorithms, than ours. How is that possible? So we are looking at animals that have a different evolutionary path since 324 million years. It is, and there's strong reasons to assume that all of these properties are not from Westlothiana lesei, the last common ancestor. But these are convergent properties, and I can argue later on why we strongly think that this is the case. So how is that possible? I will make two proposals, maybe both of them wrong, but I would like to discuss them with you. The first proposal that I'm making is that I argue that there are different cognitive ways to solve a certain problem, and they require more and more neuronal computational power, but you gain more and more evolutionary advantage by using them. But these properties, the cognitive properties that you use, are essentially similar, nearly identical, all over the different vertebrate species that were studied up to now. Let's assume here there's a rat that is hungry, that would like to eat the caterpillar. There is a cat. The rat has smelled the cat. If it would dart behind the bush and eat the caterpillar, the rat would be dead. The cat would eat it. What should a rat do to survive? One possibility is that it has sort of an inborn mechanism of that it just freezes when it smells a cat. So the cat would be the unconditioned stimulus, freezing would be the unconditioned response. It could have learned it, it could be born with it. It works. It increases the probability that this animal will survive the situation, but it's a very simple and primitive mechanism. The rat could also freeze in front of the bush, on the middle of a lawn, so in positions where the cat could easily see it. But at least it would increase the probabilities to survive to some extent. Much better would it be if the rat could learn in its lifetime a conditional discrimination task in which it learns that it has less fear as a conditioned response when there is a bush somewhere in the vicinity and where it freezes. So then still it could be that it freezes in front of the bush instead of behind the bush, but that would be an increase. But it would have to learn a conditional classical conditioning task, which is able for many animals, but not for all. Much better would be perspective taking, so the cat can't see me when I'm here. Even better would be theory of mind. So I've seen the cat eat another rat, so the cat isn't hungry anymore. I may wander around, but maybe I better stay here so I can read the mind of the cat. Now, the point is that each of these properties that I'm showing you in a literature review shows exactly the same properties, at least with invertebrates, that I could think of. Obviously, few animals are able to come up with theory of mind. More animals are able to come up with perspective taking. The majority of animals are able to learn conditional conditioning tasks, and all animals can have these very simple UCS-UCR connections. The more complex your cognition is, the higher your likelihood you survive. But the more, and finally the cat walks away, rat is free, but the more computational power you need. If you can afford more neurons, then you can think more complex. But the problem is, or the point is, at least in my literature view of these four conditions, the properties that I see between different animals is nearly identical of these kinds of cognition. Let's take, for example, classical conditioning. This UCS-UCR association, or the UCS-CS association, the time points, from aplusia to human differ from 200 milliseconds to 7 seconds. It's a very narrow time window in which they vary. So that means we do not have much variation in most of these properties within the animal kingdom. So one point, and that's what I'm arguing here, is it might be wrong that many behavioral properties that involve learning and cognition produce similar algorithmic processes of different complexities, for which then, the more complex it is, you have something out of it, but you have to pay by neuronal power. That's my first argument. Now I turn to brains and look for correlates of this. So we have chimpanzees on the one side, carrion crows on the other side, this poor lizard-like animal in the middle. And I just argued that cognitive systems are comparable between species, since most ecosystems require overlapping cognitive processes. Therefore, that might be a boundary condition. Algorithmic processes within these cognitive properties are other boundary conditions. I'm not going to talk about that because I now change my focus on the brain. I argue there are boundary conditions at the cognitive side. Ecological systems require similar processes. boundary conditions of synapses are part of every conditioning and learning process that could funnel the variation to some extent. But there are also equally boundary conditions on the neural side. I will argue that there are hard to replace mechanisms on the brain side to come up with cognitive solutions. So if the brains cannot offer multitude of implementational mechanisms, but if brains would say, we want to have that function, sorry, there is a circuit for this, and I have no second circuit available for this. So if you want to have it, I need this kind of circuitry. I'm going to argue that things like these are convincible. First, size of the brain, the pallium versus subpallium. You've seen this picture. Many years ago, we came together at Duke University and said, this is wrong, but we do not know what is the correct answer. We are not going to leave this place without an answer. And now if you look here on the left, our conclusion was the pallium is in birds as large relative as a subpallium as in mammals, and it is homologous to that of mammals without us knowing whether it is cortical or not. Could be all hippocampus, could be all amygdala, but it's homologous and it is large. That's at least step one. Step two is the size. I said bird brains are very small. How do they compute all of the properties? I just argued that they need more neurons when their brains are so small. This is a study from our Czech and Brazilian colleagues. They counted neurons. They said, forget about brain weight. Who cares about brain weight and brain size? Let's count neurons. They counted the neurons of different primates. And they all align on this blue regression. Here we have the weight of the pallium, the forebrain that is cortex, amygdala, and hippocampus equivalent. And here we have the pallium neurons in numbers, in millions, obviously algorithmic scale. And here we have the birds, the corvids and the parrots. You can see that if you have two animals with the same pallium weight, raven and three-striped monkey, the neuron numbers differ widely. Birds have 2 to 4 times more neurons per cube brain size. So they stuck in very large number of neurons. They miniaturized their brain. And they did something more. They invested most of their brains into the pallium. We just invest 19% of our neurons into our cortex. Birds invest about 70% into their cortex equivalent areas. So their evolution was about developing cognitive properties. So this can compensate to some extent the small brain size. Then we went on and looked for associative neurons. I'm not going into the details here, but these two areas, the red and the green area, these are purely associative and could be part of something like parietal prefrontal cortex. Felix Ströcken asked the question, could it be that so-called smart birds like corvids have much more neurons in these associative areas relative to the rest? Let's compare these smart birds with these, let's put it, less smart birds. We also took the ostrich as the largest bird that exists. And indeed, the smart animals have selectively more associative neurons in blue in both of these areas than the less smart animals. To the extent that the chimpanzee prefrontal cortex has 600 million neurons, and the carrion crow has in its associative areas the same number of neurons. Imagine a carrion crow that sits in front of your window, and you look at it. It has the same number of neurons in its associative areas as a chimpanzee in its prefrontal cortex. I had difficulties to imagine this. But that fits nicely. It's not about brainwave. It's about associative neuron numbers. And that fits nicely with the idea, the more complex your cognition has to go, the more neurons you have to have. But preferably, you have them in the associative areas. I'm It's approaching the end of my talk. I just talked about the prefrontal cortex. The prefrontal cortex is a conditio sine qua non, at least for mammalian complex cognition. It is at the apex of sensory motor transmission, so receives input from all associative and limbic areas, and it projects to all premotor and motor. It coordinates executive functions, weighs evidences, halts habits, restructures past stimulus response associations, holds transient information. It has classic neurons that code for task-relevant decisions that are typical for prefrontal cortex. For example, multiplexing cells with high-dimensional coding properties. We discovered something very similar in bird brains. It's a completely non-homologous area at the back of the brain. But there's so many young people who devoted their lifetime into this research that I only can show you this overall picture of them. There's too many to show individually. The same area in the NCL is multimodal with nearly, with a few exceptions, identical connectivity as a prefrontal cortex. They control the same cognitive functions, and they have the same coding properties. It's completely crazy. You have two areas that are not homologous. And whatever you look at is, I would say, to 90%, 95% identical between mammals and birds. It is not homologous. It's not the same ancestry, but it is nearly in all aspects identical to the prefrontal cortex. So it seems I'm disappointed by nature, ladies and gentlemen. I'm disappointed by nature. Whereas all of these incredible diversity that nature is producing, why is nature coming up always with the same kind of trick, the same kind of solutions? Could it be that there are not that many degrees of freedom? There are these funneling, bordering conditions that do this. There's one thing more that is lacking, or two things more that are lacking, and that's one is, and I told with some of you today during my beautiful tour through the lab, brains need feedback processes. They need a feedback about their own actions. That's typically done with dopamine. Dopaminergic systems are older than the bifurcation of birds and mammals. But the way dopamine works is identical in, I can go into the details, in the prefrontal cortex of primates and birds. They show the same differentiation across animals, birds like rats versus birds like ravens and monkeys. They show the same release properties during cognitive tasks, the same membrane properties. And so it is like a hard to replace mechanism that is needed for this. And we could even show in crocodiles that a little NCL also receives dopaminergic feedback there. So it seems to be an old solution also within the reptilian. There's one last example missing, and then I'm done and you can breathe well. The last thing that I would like to talk about is a connectome. So Mary Shanahan approached me and asked me about, could we jointly come up with a connectome of the bird brain? The interesting thing is that, and he's completely right what that is, the bird brain is so incredibly differently organized that it should not have the same kind of connectivity pattern. So what would be the connectome? What is the information flow? So we came together and analyzed this, and we came to the conclusion, I'm not going into the details here, so the connectome of the pigeon brain, we only studied pigeons, is incredibly similar to the average connectome of mammals. So also the ways information is flowing is identical. I forgot one last point, and this is really, breathe well, this is really the last. You've seen this picture before. It's the argument that they have no cortex. Well, I did not expect this to happen in my life. We discovered the cortex and the bird brain. Using completely novel techniques, we could see something that scientists about a century long could not see. It's a beautifully organized cortical structure in the sensory areas with canonical circuitries, but with the same kind of orthogonal organization as I told you beginning. But I take it personally from the birds. I take it personally. The NCL, which is the prefrontal cortex, is non-cortical. Only the sensory areas are cortical. Everything associative, everything motor is not cortical. It is as if the birds are going to say, you think it has to be cortex to be smart? Cortex is great for sensory analysis because sensory analysis needs a spatial code. You are to my left, you are to my right. It's projected crossways to my retina. All of this topography is preserved in my brain. It would be foolish to destroy it. So I'm having a two-dimensional landscape of visual input. Then I stack lamina by lamina for different analysis of the same topographic input. And I do it with somatosensory input, I do it with auditory input, I do it with visual input, I even do it with olfactory input. So the best for this is cortex. But for cognition, we came out with something better. That's something, I have the feeling that was the message, and we don't know how they do it in this NCL and related areas. So the avian sensory cortex is not the best neural, might not be the best design for complex cognition, but for sensory analysis. So this is my most beloved museum. It's in Berlin, obviously. Where else? The Natural History Museum. It's the panorama of animals. I stood there in front of these panorama for many, many years, and always when I'm there, I'm mesmerized and cannot go away. My wife says, okay, drink a second coffee, but then, please, we have to go home. Behind all of these animals, I was, in the beginning, expecting a different kind of mind. More and more, I think, there might be not that much of variation in something that we loosely call mind. It might be that their outer appearances varies incredibly, but the processes, at least at the cognitive side, within these brains, cannot be as varied because of possible boundary conditions. I take a resume. So I argue the driving force for the similarities of the neural fundamentals of cognitive system is not common heritage, but these are computational, algorithmic, and implementational constraints at all three levels. These limited degrees of freedom result in a convergent evolution of complex cognition in brain and behavior across birds and mammals. That's the only two groups of animals I really can for sure tell you this. I argue that increases of cognitive complexity, like in smart orders, goes along with a numeric increase of associative neurons, but also with a need for large prefrontal mechanisms, fast feedback systems, and an information flow system that at least resembles that what we discovered independently evolved in birds. I thank you very much for your attention.